Welcome everyone to the third webinar of the Science of Aging Series 2021, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. For this series, we've lined up a number of webinars, all focused on the science of aging being conducted by leading researchers around the world in their relevant fields. This is Sarah McFarlane from Inside Scientific, and I'm very pleased to be your host for today's event. Today's webinar is titled Experimental Muscle Mechanics in Aging and Disease, and will feature Matt Borkowski, a veteran muscle physiology technical specialist, and Dr. Rizwan Qaisar, an assistant professor at the University of Sharjah. In their presentation today, Matt and Rizwan will share some experimental protocols used for measuring muscle function and mechanics for in vivo and in vitro research. Before we get started, we'd just like to acknowledge our partners at the APS and the Alliance for Aging Research, and also to thank the sponsors for this webinar series for making it all possible. So without any further delay, I'm very pleased to welcome Matt Borkowski to the floor. Matt, take it away whenever you're ready.
Thank you so much, Sarah. So Aurora has been serving the muscle mechanics community since 1997, when we began selling only instruments for force, measurement, and positioning motors. And since then, we've tried to partner with the research community by further developing stimulators, software, experimental apparatus, and packaging them into turnkey systems. These test systems range from testing isolated single cells and fibers, all the way up to testing forces produced by whole large animals, such as rodent models, dogs, pigs, and even sheep. We'll talk a bit first about the higher end of the spectrum and then spend a little bit of time looking at our system for single fiber mechanics. So let's first discuss this whole muscle, whole animal system that we unofficially call our 3-in-1. The first experiment that I'll describe is to surgically remove the muscle from the mouse or animal, place it into an oxygenated bath chamber to keep it viable. The muscle is stimulated with field electrodes, and the force produced by that one muscle can be measured along with simultaneous changes made to the muscle length. This is what we would term in vitro. The term ex vivo has also been used for this type of experiment. A similar experiment, and the next one we'll take a look at, is isolating a single muscle, however, doing it what we would call in situ. And there's a greatly reduced amount of surgery required. This is performed by making a small incision in the animal's leg to expose one of the muscle tendons directly. The tendon can be cut and sutured directly to the transducer. We're performing a very similar experiment to in vitro. However, we have the advantage of not having to remove the muscle from the body. There's no bath to keep the muscle viable because the anesthetized animal is keeping it viable with its own blood supply, perfusing it that way. The final experiment we term in vivo. This involves anesthetizing an animal and measuring the aggregate torque produced by either the entire plantar or dorsiflexors of the lower limb. There's no surgery required. The attachment point is a small shoe, which the animal's foot is taped to, and the contraction is produced by small needle-style electrodes. The experiment is fully intended to be non-terminal. Now that we know a little bit about what experiments the system is capable of performing, we can take a closer look at how the system works and at each component that plays a role in its versatility and its power. The system is sort of based around our flagship instrument, the dual mode lever. So dual mode means that the lever system is not only a force transducer, but also a position transducer as well. And so in addition to being able to measure those two variables, it can control either one of these parameters while simultaneously measuring the other. And it's able to flip instantaneously between which parameter is controlled and which parameter is measured. Since the instrument performs both measurements in one, it only requires one attachment point. Not only does the one attachment point save time and it minimizes artifacts, it also enables the in situ and in vivo experiments to be performed. Depending on the experiment, either a lever arm or a foot pedal is tightened onto the shaft of the system. And because the instrument is not simply a force transducer, it's capable of performing far more complex protocols than simply measuring maximal isometric strength. Both eccentric and concentric contractions become much more trivial, along with measuring power, force velocity, work loops, and many other protocols. Essentially, we can use the system to try to better model how a muscle works in real life. So now when we say one attachment point, let's take a look at what these experiments look like in more detail. So to briefly describe the process for an in vivo experiment, the foot of the animal is taped into a boot, as I mentioned, and it sort of acts as a lever that attaches to the dual mode instrument. Direct nerve or direct muscle stimulation will elicit a contraction, either of the aggregate plantar or dorsiflexors of the lower limb. Usually these are the ankle flexors. The torque about this ankle joint can then be measured. So we can create a variety of experimental conditions that model real-life exercise or damage to the muscles by varying what types of contractions are applied. Mimicking exercise protocols can be accomplished as well by setting the isotonic resistance of the pedal and then performing a battery of concentric contractions. You could model a downhill running protocol to induce damage by applying eccentric contractions. And these are just a couple examples. We'll talk a little bit more about some of these contractile experiments later on. So a bit about the pros and cons of in vivo. It's for sure the most high throughput assay. And it's across the ankle joint, which is, of the three, the most physiologically relevant. There's no surgery required, and we don't interfere with the vascular supply of the muscle as well. And of course, the big one, repeated measurements and longitudinal studies are possible. So it's a little bit of a challenge to determine exactly the individual contribution of each muscle, and of course, this is for lack of a better term, a bit of an acupuncture technique in placing the muscles. If there are the electrodes, rather, if there are problems with the neuromuscular junction, it can be a bit of a challenge to elicit a consistent contraction. So now on to in situ. An in situ experiment allows this the researcher to sort of isolate a muscle of interest, but you're keeping one or both of the ends of the muscle undisturbed, so as adequate blood flow is maintained. So the most common muscles that are used for these tests are still the ankle, plantar, and dorsiflexors, and this is mostly because of accessibility to the tendons. However, all sorts of other muscles, trapezius, vastus lateralis, even masseter, have been tested. This experiment is almost always terminal, as the tendon usually must be surgically cut to attach to it. However, there are some instances where this has been performed as a survival study. Okay, again, now what are some of the pros and cons of these two systems? It is more surgically invasive. However, the vascular supply is still preserved. You're only isolating one muscle, meaning that you can measure any given muscle's specific contribution to contractile performance. It is a bit invasive. It's certainly technically more difficult, I would say, than the in vivo case. And it really is challenging to perform this, though not impossible, as a survival study. Okay, finally, the in vitro, or the classic experiment that I would say the majority of physiologists are probably familiar with. The muscle, of course, is surgically removed and placed into a temperature-controlled and perfused chamber. This configuration of our system allows the researcher to sort of place this muscle in the chamber or bath. You attach to a fixed post at one end and our dual mode system to the other. And what's really nice about this dual mode lever is that you're able to control both force and length, and you're only attaching to one point of the muscle. Complete characterization of the muscle is now possible with only this one attachment point. So you're able to perform a range of protocols, as I mentioned, including lengthening contractions, isotonic contractions, concentric fatigue protocols, and length velocity. It becomes much more powerful. So now, what are some of the pros and cons of this last one? You're making a measurement of muscle contractility that's independent of the nerve, which is not so of the other two techniques. If you're applying field stimulation, I should qualify that. So you can still dissect the muscle with a portion of the motor neuron attached and bypass a field stim, and this technique is desirable if you want to load the bath with some sort of drug or compound. Of course, as soon as the muscle is removed, there is no bodily blood flow keeping this muscle viable. You've got to use some sort of oxygen perfusion into the media, and this is limited by diffusion. It is a challenging technique, not insurmountable by any stretch of the imagination, but it does take a bit of practice to perfect. And of course, this is certainly a terminal study. So just moving along here, I keep alluding to this concept of completely characterizing muscle function. We've touched on this notion, and I've stressed that this instrument with one attachment point makes it much easier. So we're leveraging the function of this instrument to make this or permit a battery of contractions that can be applied across all three techniques, which is unique. But what does it really mean? And essentially, it means that we have the flexibility and freedom to model all sorts of contractile activity and paint a very full picture of functional performance. There are many cases where a basic assessment of isometric strength is sufficient to observe differences in functional performance, but it's not always painting a full picture. So just a quick little table here of the different contraction types and a quickie comparison of when you might consider them. As with all experimental design, the choice of how to model a contractile test, it's not perfectly cut and dried. The goal of the functional test must first be considered before choosing the experiment type. This is the quick overview here. I mean, generally, isometric contractions are best for making an assessment of maximal strength and looking at some temporal characteristics of contraction. Eccentric protocols are typically used for contraction-induced damage or to try to look at hypertrophy. And concentric or shortening contractions are typically used to look at work, force velocity, the power of the muscle, and so forth. Attempting to model everything in one experiment is rarely feasible or productive. So it really boils down to considering the goals of the functional test before choosing this experiment. Now, with that all said, let's have a look at the lower end of the spectrum, the single fiber system. And when I'm talking about a system for single fibers, I'm specifically referring to a system for single permeabilized muscle fibers. Before I get into the protocols and some of the information about the system, let's just take a moment to define what I mean by permeabilized fibers. So in the simplest of terms, permeabilized fibers, they've been made permeable to the flow of calcium ions. And how is this accomplished? Typically, a biopsy is dissected into some workable bundle, and these bundles are immersed into chilled permeabilizing solution, which makes the membrane porous. Depending on the size of the bundles, this can take a few minutes, up to maybe an hour. You can also remove the membrane of the fibers by mechanical means, if you imagine pulling a sock from your foot. But this is less common and much more difficult. Once permeabilized with the solution, they need to be washed to remove any of this permeabilization agent, and they're stored in a storage solution overnight, chilled. You can generally keep these samples for longer periods at minus 80 degrees in a different type of storage solution. So the next day, we'll tease out individual fibers from this small bundle, and we mount them in the apparatus for testing into a relaxing solution, and then we create various solutions for with varying levels of calcium for contraction. Okay, how do we actually mount or handle these fibers? Once you dissect it out of the bundle, it should be transferred into a larger mounting bath of relaxing solution. It's quite small. They're difficult to handle. People use something like a cut pipette tip as a transfer vessel, so to speak. And then there are two main methods of attaching onto these fibers. Aluminium foil clips, which sort of act as a crimp to hold it in place, or directly suturing onto pins. In some cases, adhesives have been used, UV-based glues, silicone glues, shellacs. This is a little bit less common than it used to be, but all of these methods sort of depend on exactly the type of fibers being used and their size. And this is really a critical portion of the experiment. If it's mounted improperly, it'll slip out and the data will be unreliable. Okay, so a bit about the instruments that we need to make these measurements. So first, of course, we need a very sensitive force transducer. Typically, we pick one in the range of 5 to 10 millinewtons maximum. Absolute forces generated by these fibers are in the hundreds of micronewtons, maybe up to a millinewton or two. This all depends on size. And so the preps are not long. They're typically, perhaps less than a millimeter, maybe up to a few millimeters. So any compliance in the force transducer will mean that we're not making a measurement that's truly isometric, and we'll be underreporting the maximal forces. We also need some sort of fast servo motor or length controller, you could call it, to make changes in fiber length. You typically need at least hundreds of microns of movement capability to be able to make slacks or steps of fibers of this size. These motors are also, they have a good frequency response, so they're useful for making stiffness measurements as well. The other part of the system is this automated 8-well chamber, which can automatically index between relaxing and calcium solution. This is software controlled, and of course there's a whole integrated package for making standard measurements. So just very briefly, what are some of these measurements? Well, the first is the standard Force Pca. This is done isometrically. We hold the fibers at a constant length, and they're immersed in different solutions of varying calcium concentration. And the force output is then plotted in this sort of sigmoidal shape. This is sometimes referred to as calcium sensitivity as well of the fiber, rather than Force Pca. The next one is a KTR experiment, coefficient of tension redevelopment. In this test, the fiber is immersed in very high calcium solution after pre-activation. So it produces a maximal force output immediately. And once this plateau has been reached, the fiber is rapidly slacked by a great deal and then quickly restretched. Tension begins to redevelop, and the time it takes to begin redeveloping tension can be measured. A sort of somewhat similar protocol called the slack test is often performed in skeletal muscle fibers without the restretch to determine the unloaded shortening velocity. And these experiments can sort of help us understand the contribution of the cross-bridge cycling and the mechanisms of the thin filament, such as how something may affect the binding of calcium to troponin or the interaction between the actins and myosins. Finally, the last experiment here is what we call force velocity. We maximally activate the fiber again in a high calcium solution. We hold it at a constant length, and then this is sort of recorded. Once relaxed, we activate it again, and once the plateau of force is reached, we apply this series of descending load clamps, and this is done with a fast feedback loop in software. And what essentially happens is that we control the force, we hold it at a constant value, and we're looking at the shortening of the fiber. So the product of the load and the shortening velocity at any given point essentially yields power, and we can plot this power curve against the relative tension of the fiber. And this sort of gives us some indication of fiber performance. This is routinely done in whole muscle as well. But by using single fibers, we can sort of focus on questions about the myofilament, again, such as interactions between actin and myosin. So finally, why bother using these techniques for aging research? I think a big one is that it allows for longitudinal tests that monitor function as the animal ages. Some of these more in-depth properties, temporal properties and kinetics of contraction, sort of going beyond just looking at maximal strength, can be really well quantified. In an in vivo model, we can potentially use this as a training tool or an exercise intervention. The single fiber techniques allows any species to be used, and that includes humans, which I think is certainly of interest. And all of these techniques sort of remove the voluntary component of contraction, and it really only focuses on neuromuscular performance and basic contractile mechanisms. So there's no need to train the test subjects, train your animals to have them run or grab something. It completely bypasses that. So I think when all of these things are considered, these are potentially very powerful tools for sarcopenia, aging, and gerontology research. Thanks so much. Toss it over to Rizwan here now. I'm sure we're all eager to get started with his presentation.
Okay, thank you, Matt, for that fantastic presentation. And now I'm very pleased to welcome Dr. Rizwan Qaisar to the floor. Rizwan, take it away whenever you're ready.
Thank you, Sarah. So this is a very general snapshot of sarcopenia, how it occurs, what it is, what are the molecular mechanisms, what are the possible interventions. And I'll discuss some of my data from my PhD, which ages ago, and my postdoctoral work and the later work that I did in Europe, US, and my ongoing work here in United Arab Emirates. I guess everybody knows what sarcopenia is. It's the age-related muscle loss, and it's quite eminent here. You see the CT scan of mid-thigh of a young and old person, and you see that there's quite prominent muscle atrophy and infiltration of fats with aging. When does it start? It starts around about in the late 20s or early 30s, and it goes on and on. As the age advances, there is an accelerated muscle loss. It's 15% muscle loss per decade starting 70 years of age, and it's slightly less before this. The red zone is this period. So when we are in this zone, which is about 70 or 75% of muscle mass, our lifestyle becomes dependent, and then we need help in our everyday tasks. This is the loss of muscle mass, but there is also a concomitant loss of muscle strength and muscle power. And for the muscle strength, it's about 1 to 2% per year. And muscle power, which is a measure of strength and how fast we move our muscles or the speed of contraction, is slightly more than the loss of muscle strength, which means that generally our movements are sluggish, and there is more chance of losing balance, fall, and fall-related injuries, which further compromise our sarcopenia phenotype. So there are various definitions of sarcopenia. These are the four more common ones. The more common one is this one, the European Working Group on Sarcopenia in Old People. What this does and what the other definitions do, they generally consider the muscle strength, muscle mass, and the physical performance. So if we are losing muscle strength, the muscles are becoming weaker, they are atrophying, and our physical performance, which is generally measured by gait speed, it's getting low, or our gait speed is getting slower. These are some quite good indicators of sarcopenia. So what EWGSOP2 criteria looks at is the muscle strength, and usually it's measured with hand grip dynamometer. So we measure the strength of hand grip muscles, and these are the cutoff values for men and women for sarcopenia. If that's not available, the chair stand test can also be done. For muscle mass, there are scales available to measure the body composition, and they are pretty cheap. So they measure the appendicular skeletal mass, and these are the values. A better measure is to divide the ASM, or appendicular skeletal mass, by the height square, and we get these values. So these are the cutoff for sarcopenia. Of course, it's not just the strength and mass, but we want to look at the physical performance of the people. And a very common indicator for this is the gait speed. So if gait speed is less than 0.8 meter per second, that's pretty good indicator of well-developed sarcopenia. These measures are easy. They can be measured easily in the clinics. So there are hand-grip dynamometers, commercially very cheap. I think it costs like $20. Then there are scales to measure the body composition. Similarly, the gait speed can easily be measured, and they give us a pretty good idea of the risk for sarcopenia or diagnosis of sarcopenia as well. Another common measure that I use a lot in my studies is SPPB, or Short Physical Performance Battery. It hardly takes like 5 minutes. So what we do is that we look at the chair stand ability of the patient, their balance, their gait speed, and based on these three measures, we assign them scores. So below a cutoff score, which is usually 8 out of 12, that's a good indication of sarcopenia. This could be something for an early assessment, and then, of course, once we identify the high-risk patients, we can go for more thorough assessment of this. And here's a very common scheme to measure or to diagnose the sarcopenia. It can develop early, but usually the patients are like 60 plus, 65 plus. So first we measure the gait speed. If it's less than 0.8 meter per second, then we look for, actually I should be here. So if it's less than 0.8 meter per second, we look for muscle mass, we look for muscle strength. And if we see a reduction in all three parameters, that would mean sarcopenia. This is for clinics, this is for humans, but in lab, we can also test it in mice. And there are many mice models of sarcopenia which show an accelerated aging or accelerated muscle loss. And they are mostly genetic, and they target different signaling pathways or different organelles in the mice. It could be mitochondria, it could be more inflammation, it could be blocking of apoptosis, autophagy, disruption of calcium handling, less anabolism, or so and so. The one model that I have worked a lot in Dr. Holly Van Riemen's lab is actually, I'll show it here. So that's the SOD1 knockout mouse, and SOD1 we know is the superoxide dismutase, which is an antioxidant enzyme. So once this is knocked out, the mice developed a high oxidative stress, which could be global, or we can do it in an organ-specific manner in the muscles or in the motor neuron and so. And just to show that there are different advantages and disadvantages of these mice models, of course, they don't mimic humans completely, but mice have a shorter lifespan compared to humans, so we have to wait less for the mice to get old. If we want to try interventions to prolong lifespan or health span, it's easy to measure those and see their effects compared to humans. The disadvantage is that not all the phenotypes of aging and the sarcopenia are recapitulated in these mice, and of course there is some genetic manipulations in those, so it may be that other body systems are also affected, and then their detriment affect the muscle health or sarcopenia phenotype. So what I have been working a lot on is on the SOD1 knockout mice, and so here are the mice. This is actually Dr. Holly Van Riemen's group where I worked on these mice. Here we have a wild-type mouse, and here we have an SOD1 knockout mouse. Both are 10-month-old. 10 months is just about adult age for mice. But if you look at this mouse and then look at its muscles, we will see quite striking atrophy of all the hind limb muscles. These mice, they develop muscle atrophy, muscle weakness much earlier than the age-matched wild-type mice. And I'll come back to I'll show some data from these mice, so I'll come back to those. So there are different ways to measure the parameters of sarcopenia. Of course, first thing is to see if the muscles are stronger or they're weaker. And in vivo, we can measure it in humans with the hand grip dynamometer. We can also measure the knee torque with the knee dynamometers. In mice, there is this equipment by Aurora Scientific, which measures the in vivo hind limb muscle strength. This is in live animals or humans. But if we get the biopsy, we can look for muscle mass, of course, in biopsy to see if there's an atrophy or no. And we can do the single fiber mechanics. So we can look for the single fiber force, and then we can do different imaging techniques on the biopsy tissues. And what I have done a lot is the myonuclear domain and the myonuclei and their organization. I'll briefly show some of my data from there. And also the neuromuscular junction, the cross talk point of the motor nerve and the skeletal muscle. Other than this, the enzyme activity that I've been doing a lot is the Sarca ATPase. This is like my enzyme of interest. This enzyme is in the Sarcoplasmic Reticulum. It's involved in the uptake of calcium following contraction, and its activity is highly, highly compromised in aging, in sarcopenia. So I have been interested in this, and I believe that this is a pharmacological target to prevent or reduce aging as well. And other than this, of course, we can do like routine biochemistry techniques to see the expression of different proteins and different like mRNAs as well. One thing that I've been lately doing is to collect blood from humans and look at different serum biomarkers of sarcopenia. And I'll show some data from these as well. So these are different ways that we can assess the muscle in aging in sarcopenia. This is the single fiber recording. This was a major part of my PhD thesis about like 10, 11 years ago in Lars Larsson's lab in Sweden. So take a single muscle fiber, attach it to two connectors, and then expose it to different calcium concentrations, and then see how it contracts. So here is a relaxed fiber, here is a contracting fiber, and just look at the length change and the force development at different calcium concentrations, and from those we can see how strong or weak this muscle fiber is. Okay, of course we do it in single muscle fibers, but in mice we can do it in whole muscles also. So this is in situ mouse gastrocnemius muscle recording. So here what I do is that I cut off the tendon of gastrocnemius, remove the plantaris and soleus, which are deep to it, and then attach the tendon to a force transducer, and then electrically stimulate it and measure the force. The good thing here is that the nerve is intact. So I can place an electrode on the nerve, I can place an electrode on the muscle and stimulate them and see if the force from nerve stimulation is different than force from muscle stimulation. And this is like an indirect measure of the neuromuscular junction, or how good it is in passing on the signal from nerve to muscle. And also there's an in vitro recording also. It's not possible for big muscles like gastrocnemius, but it can be done in more thin muscles like EDLs and soleus also. And these are again the Aurora scientific instruments. So back to sarcopenia. So sarcopenia is age-related, so it's physiological, it's primary, and many things happen in muscle with primary or physiological sarcopenia. There's more oxidative stress, more denervation, more inflammation, more degradation of proteins, and many other things. They also contribute to this age-related sarcopenia. The hormones are less, the protein synthesis is blunted, less vascularization, and so on. So sarcopenia can be accelerated in different age-related diseases and conditions. The sedentary lifestyle, bed rest, different diseases, reduced nutrition, malabsorption, all of them, they speed up or accelerate the progression of sarcopenia. So this is secondary sarcopenia, which is more pathological, and this is preventable as well. Because if we can treat the risk factors, we can at least reduce it. And among those diseases, these are some of the diseases of interest that I have been working on, COPD, and then I have patients from heart failure, patients with Parkinson's and different other neurodegenerative diseases. And of late, I have some COVID-19 patients also, and I'm looking at the muscle decline in them which seems accelerated compared to age-matched healthy people. At the molecular level, many things happen in sarcopenia, in skeletal muscle. Like one can spend like hours and hours describing them, but in general, there is an imbalance of protein synthesis and degradation, less synthesis, more degradation. This contributes to sarcopenia phenotype, and then there are many pathways which are activated or blunted, which also lead to muscle atrophy, weakness in sarcopenia. From here, I'll just describe some of the changes in muscle that I have studied, and I'll just focus on these organs, these organelles in the skeletal muscle, the myonuclei, the neuromuscular junction, mitochondria, the sarcoplasmic reticulum, and the muscle fiber types and the myosin isoforms. About myonuclei, generally the procedure is pretty same. We have a muscle biopsy, we take single muscle fibers and attach it to two connectors and take it to a confocal microscope. And this one is stained with DAPI and rhodamine for actin, DAPI for nuclei. And with confocal, one can do the 3D scans of the muscle fibers, and they look a bit like this. So the red are actin and the blue are nuclei, and they are a bit like this. So we had a software which could analyze the 3D positions, the XYZ coordinates of each myonucleus in the muscle fiber, and they would look like this. And generally the journals, they like colors, right? So this one went on to the covers of these journals also. Doing this technique, we showed that the nuclei count per unit length is not changing much with aging. So nuclei, they resist apoptosis with aging. They do apoptose, but like it's quite less compared to the amount of atrophy of the whole muscle fiber. And since the atrophy is there, but the loss of nuclei is not parallel, it's much less. So the myonuclear domain size, which is the cytoplasmic volume per myonucleus, that's reduced with aging. But the important thing here is that even though the myonuclei are not lost, their organization is messed up. So here is a young muscle fiber. You see that the myonuclei are quite evenly spaced. But when we look at an old muscle fibers, we see lots of clumping of myonuclei. So there are wide open areas, and then there are many, like 3 or 4 myonuclei together. Then there's again an open or bare area, and then there's another clump. So the organization of myonuclei, this is affected in aging. It's also affected in denervation and some other diseases. And we believe that this is an important contributor to reduce transcriptional activity, reduce protein synthesis in sarcopenia. So the other thing that I have been really studying a lot is the neuromuscular junction. This is the interface of nerve and muscle. There are many factors which can affect the NMJ or neuromuscular junction, like systemic inflammation, the degeneration of motor neurons, and oxidative stress in motor neurons. They all affect it. And we have this mouse model, the SOD1 knockout mouse model that I showed before. And using this mouse model, we have deleted SOD1 specifically in the muscle only, or in the motor neuron only, or both in muscles and motor neuron. And our question was that which one of these two, the muscle and nerve, which one of these two is the major contributor to neuromuscular junction disintegration in sarcopenia? And it seems that the critical elements for maintaining a good and healthy neuromuscular junction, that's either nerve alone or both the nerve and muscle together. But apparently the contribution of muscle to a healthy neuromuscular junction, that's not much compared to the contribution of nerve. So this was the morphology of the neuromuscular junction. It's disrupted, and we see that at the muscle side, the acetylcholine receptors, they are fragmented, they are dispersed. It's quite a characteristic finding or feature feature of aging. There's also reduced overlap between the motor neuron and the muscle with aging. So these are the structural changes or morphological changes in the NMJ with aging. We also find many functional changes which reflect the structural disorganization. And like I said before, for example, in gastrocnemius muscle, we can do an in situ recording. We can place an electrode on the muscle. We can place an electrode on the nerve, stimulate them, and see if the force recordings from the two stimulations are comparable or no. And these are some of the force recordings. So the blacks are the muscle force due to nerve stimulation. The gray are the muscle force due to muscle stimulation. Generally, we see slightly less force with muscle stimulation compared to nerve stimulation. Just notice that as we age more and more, the difference between nerve and muscle, it's blunted. It's gone. And this shows that something is happening to neuromuscular junction, and it's not very efficiently transmitting the signal from nerve to the muscle. And we can actually measure the difference between nerve and muscle here. So generally, the difference is in minus, which means that the nerve stimulation gives a stronger force compared to muscle stimulation. It stays minus, but in advanced age, the difference is plus, which means that the nerve stimulation gives less force compared to muscle stimulation, which means that the neuromuscular junction functionality is affected and is not very efficiently transmitting the signal to the muscle. So there is disruption of function, disruption of structure of the neuromuscular junction with sarcopenia. A lot happens to mitochondria also with sarcopenia. Generally, we know that in a healthy muscle or in any tissue, there is this fission and fusion going on, and then there is mitophagy of old mitochondria, biogenesis of the new ones. What we have found in our studies is that with aging, there is more blunting of fusion. So we don't see much fusion. We do see much fission, but not much fusion. The biogenesis, of course, is also reduced. And as a result, we see reduced mitochondrial function, which means that they are making less ATPs and they are making more ROS or reactive oxygen species. So more ROS we see, we also see the consequence of it. There is more damage to DNA, more damage to proteins, more peroxidation of lipids, all because of mitochondrial dysfunction. Mitochondria are also producing less ATPs, their VO2 max is reduced, their respiratory capacity is reduced, and these are like pretty well established findings in sarcopenia. This is actually my data. So I used to isolate mitochondria from the mouse tissues, and I've done it from humans also. So they can be isolated, and then their peroxide production and their ATP production can be measured in different states. Generally, with aging and with different muscle diseases, they produce more peroxide and less ATPs. So this happens with them. Other thing that I have studied a lot is the sarcoplasmic reticulum. This is actually one of my latest love in sarcopenia. So I'm really interested in the calcium regulation and calcium homeostasis and how this is disrupted in sarcopenia. So as we know, sarcoplasmic reticulum is the major reservoir of calcium during muscle contractions. It has ryanodine receptors which release calcium for contraction, and it has sarcopumps which take the calcium back after contraction. So these are some of my papers on this, and I have shown that there is disruption of calcium homeostasis, calcium handling by the sarcoplasmic reticulum, which seems a major contributor to sarcopenia phenotype. In mice, and also I have some data from humans as well. So to summarize about sarcoplasmic reticulum, on the calcium release side, the calcium release channels, like we have shown that with aging, there is more oxidation of ryanodine receptors, And there is this very interesting protein, FKBP or calcitabin, which is attached to the ryanodine receptors. We have shown that it is detached or dissociated in aging. The result is there is more calcium leak and there's less calcium available for contraction. On the uptake side, we see there is nitration of sarcoproteins, there is reduced sarca activation, and reduced calcium uptake after contraction. And all these lead to reduced calcium sensitivity of the sarcomeric proteins, and which results in muscle weakness and sarcopenia phenotype. This is sarca pump activity, which we used to do and we still do in the lysates of the muscles. And we can see that the 16-month-old mouse, which is this one, it shows higher activity than the aged mice, like 26-year-old mice. So sarca activity is reduced, it's compromised with aging. And then there is this compound, CDN1163, which is an allosteric activator of sarca, and like this has been a major focus of my studies, and I show that it can prevent or reduce the sarcopenia phenotype, and I'll show some data from this compound also. Other thing that everybody knows pretty well with aging, with sarcopenia, is that there is preferential loss of type 2 fibers. These are all the muscle fiber counts, and they, like muscle fiber count reduces with aging, and generally the proportion of type 1 fibers increase and type 2 fibers decrease, because type 2 fibers are preferentially lost, and we can measure them with this STS page. Why type 2 fibers are more susceptible to age-related muscle loss? And there are many problems, many structural problems in them. Their Z discs are thin, their tight ends are smaller, they develop more force, their dystrophin, which is which buffers the force towards the cell membrane, they are also smaller in type 2 fibers. So type 2 fibers, they have more wear and tear, and they are degenerated first in sarcopenia. There are many interventions, and I think it's like textbook stuff. So we know a lot about the sarcopenia interventions, the physiological interventions like exercises, caloric restrictions. They are pretty well known. They are nutritional interventions for those people who are bedridden or who can't do the exercises. There are hormones, especially in women after menopause, like estrogens. And then in men, there are growth hormone and testosterone. They have their advantages and disadvantages. Like I had some, like long time ago, I had studied in monozygous twin pairs, females, which were either, like one of the twin pairs was on HRT or hormone replacement therapy, and the other was not. So we showed that HRT is quite beneficial in protecting muscle mass and strength during aging. And then there are pharmacological therapies, the myostatin blocking antibodies. They are in clinical trials. They are ACE inhibitors. And then there is CDN1163, which is something I have been working a lot. And it's a sarca activator. And I have shown that with the restoration of sarca activity with CDN1163, there is prevention of sarcopenia and age-related muscle mass and atrophy. So for CDN1163, we used to inject the mice with inject them or put it in food. And what happens is that CDN1163, it restores the sarca activity. So the age-related compromise of sarca function is gone. And this is associated with the prevention of atrophy of the gastrocnemius muscle mass and the prevention of force or weakness of the EDL. So, I mean, there are many interventions in literature which show prevention of weakness, but like restoration of muscle mass is quite rare in literature. So this is something really specific or special about this therapy. And so there are many biomarkers of sarcopenia, and they relate to autophagy, apoptosis, growth factors, neuromuscular junction, and there are several actually. Some of these they spill in blood and in urine, and they can be measured there, and they are quite useful in the in assessment of sarcopenia. One of them that I am really interested in is this one, the CAF, or C-terminal agrine fragment. This is actually a fragment of agrine at the neuromuscular junction. There are actually two fragments, and this is one of the two fragments, the larger fragment, actually the smaller fragment, the CAF22, the larger is CAF110, and this one is spilled in blood, and its levels can be measured, and I have shown actually in past 2 or 3 years, and there are several studies now, that CAF22, it relates, its blood levels, they relate with sarcopenia phenotype pretty well. For example, we see that in aged humans who are healthy, its levels are like this, but in aged people of the same age but with different age-related diseases like COPD or heart failure, the CAF22 levels are increased in the blood. So there is this spilling of this, which probably means that there is disintegration of neuromuscular junction, and then this marker shows up in blood. Its blood levels, they show pretty good relation with the hand grip strength, so they are a pretty good measure of how strong the muscles are. Also good measures of ASMI, or appendicular skeletal muscle index, or the muscle mass, and generally in weaker muscles and weaker muscles and the atrophied muscles, we see that the CF22 levels are higher in the blood. And I've shown also that the SPPB, which is the measure of physical activity, those scores also show pretty good relation with the CF22. So generally, people with lower scores, they show higher level of CF22, although it's not pretty robust, but there is some relation with the physical activity. And the last thing, that's my ongoing project, and is the unpublished data, is the COVID-19 patients. And there are some studies which show that the COVID-19 and the muscle, they have pretty good crosstalk going on. So people with sarcopenia, they are more prone to developing COVID-19 and developing a more stronger phenotype, while people with COVID-19, they are prone to having a sarcopenia phenotype or more muscle weakness and atrophy. So I had these patients, and so I showed the length of the duration of disease of COVID-19 in days, and this counts from the first PCR positive test till the PCR negative test. And we see that the patients who don't have sarcopenia, the duration of disease is generally low. Patients who start with sarcopenia, the disease is pretty longer in them, while the patients who develop sarcopenia during COVID-19 infection, again, the duration of infection is much more than the patients without sarcopenia. So there is some correlation between the length of COVID-19 infection or the duration of COVID-19 disease and the sarcopenia phenotype. I looked at the CF22. There's not very robust finding, but it does show that the CF22 levels are slightly more in the COVID-19 patients. Actually, I had blood level, the plasma stores from those patients before they develop infection. I had actually a large cohort of patients, and some of them developed COVID-19. So I just analyzed their plasma levels before and after the infection. And there are some indications that the CF22 levels are higher following the COVID-19 infection, which might mean that the COVID-19 is affecting the neuromuscular junction in them also. And the CF22 levels also show a pretty good relation with the duration of disease. So higher CF22 levels in plasma would mean a longer duration of disease or delayed recovery from the disease. So there is some relation between sarcopenia phenotype and the COVID-19, at least in the elderly. So just to summarize, so we know that sarcopenia is loss of muscle mass and strength with aging. It's physiological, but different pathologies will accelerate it, and it becomes pathological in those cases. There are many changes in muscle at the cell level, at the organelle levels, and at the molecular levels. And sarcopenia is it aggravates almost like, or it's related to almost all organs, and it's related with many other diseases. So there are many interventions going on to reduce the rate of sarcopenia, and there's some evidence that sarcopenia may aggravate the other age-related diseases and the COVID-19 also. And the crosstalk is bidirectional. I wanted to post a photo of some sarcopenia model, and I found this actually. So this would be me like in 30 years or so, something. So I would like to thank all these like these guys. So Lars Larsson from Uppsala was my PhD supervisor. Roberto Bertinelli, I did my first postdoc with him in Pavia in Italy. And Holly Van Raman in Oklahoma. I did my second postdoc, and then I was a faculty member there. And nowadays I am in University of Sharjah. I don't have a photo of my group. My group is actually just 3 people, including me. So I just posted the photo of the university. There are many grant sources and funding sources, and I'm really thankful to all of them. So thank you very much.
Okay, fantastic. All right, so I'm going to just give you a quick reminder that there is an Ask a Question panel there. If you haven't already used it, it's now wiggling on your screen. So send us any questions that you have for Matt and Rizwan, and we will address as many as we can. I know we're a little bit over time here, so if you can stay, that's fantastic. If you can't stay, before you go, we would love to get your feedback on the webinar. So please fill out that survey at any time. All right, so Matt and Rizwan, do we have you back with us?
I'm here, Sarah. I'm ready for the questions.
Yes, same here. I am ready.
Okay, fantastic. All right, so our first question is for you, Matt. Has any more work been done to adapt the Aurora scientific device for non-human primates?
Not a tremendous amount. As you can imagine, it's not the primary animal model being used with the 3-in-1 system. The model of platform that we use for non-human primates. We've made some changes to the clamps, but that's mostly to accommodate larger animals and more like porcine models. That's probably the biggest change. So there's not a tremendous amount to report there.
Okay, great. The next question is for you, Rizwan. This question is, are you aware of any pharmacological or nutritional interventions that specifically increase muscle speed and power, knowing that mass and strength decline rapidly, but not quite as fast as power?
Not really. I mean, there are so many protein supplements that increase the mass, and they mostly affect type 2 fibers. And type 2 fibers, they are pretty flexible in terms of size, so they can hypertrophy, they can atrophy like relatively easily compared to type 1 fibers. So if we do see a change in type 2 fibers, it might like translate in a strong, like a faster speed of contraction also. But like, so that would be an indirect effect on the speed. I don't know if there is a supplement, a nutritional supplement, which could directly or like affect the speed.
Okay. Sorry to interrupt. I just wanted to jump in. I am aware of one recent publication where our system was used, and they looked at the effects of dietary nitrate supplementation, and this was in mouse diaphragm muscle. In elderly animals, and they showed that there was an increase in shortening velocity and an increase in power. This is Dr. Leo Ferreira's group, and he published this in 2020 Journal of Physiology.
Okay, great. We've got another question here. I think this one is for Rizwan. Does oxidative stress induce sarcopenia? And if so, what could be the key free radical involved?
Okay, so this is a ongoing debate. Like there would be papers titled as "Is oxidative stress a cause or consequence of sarcopenia?" and it still goes on. So I mean, there are studies which show that if you could, and we have the mouse model also, the SOD1 knockout mouse, which show that if you could reduce the oxidative stress, you improve or you reduce the sarcopenia phenotype. So like there is less atrophy, less weakness, and so. What free radicals are primarily involved? I can't really say any specific one, but like specifically, I mean, it is associated with sarcopenia. It has definitely contribution to sarcopenia. Is it the main player? We don't know. The other thing is that we have to talk about oxidative stress in what organ? Is it muscle? Is it nerve? Is it both? Is it global? Apparently, oxidative stress in muscle alone, it's not like a hammer. So it does cause some damage or detriment, but it's not as much as an oxidative stress in the neuron. So it has a contribution, but we don't really know if it's the major player or it's a cause or it's a consequence.
Okay. Thanks for that answer. We've got another question here. I'm going to combine a couple because we've got quite a few of a similar question. So this question is, have you done any CDN1163 trials in humans, like clinical trials? And if so, what are the side effects?
I haven't done it. And one reason is that it's extremely costly. And the other thing is that for short term, like in mice, for short term, like for a few weeks, we don't see an effect. To see an effect, we have to administer it to mice for like many weeks. If you translate this to humans, that would be CDN1163 apparently for years and years, and it's so costly that we can't afford it. People have done it in mice for different organs because like all the organs, they have endoplasmic reticulum, which is sarcoplasmic reticulum in muscle, and they have those Sarca enzymes. So CDN1163, it improves the Sarca enzyme activity, and it has positive effects against neurodegeneration, against metabolic syndrome, against like bad things going on in liver and pancreas. But for now, as long as it's costly and as long as its effect come after like a chronic dose, I guess it will stay to the mice.
Okay, fantastic. Just in the interest of time, I'm going to make this the last question. This question is, would the increase in CAF22 and the resulting impact on the neuromuscular junction in COVID-19 patients could it be a result of the lack of exercise and movement caused by the hospitalization?
It might be actually, but some of our patients, they were ambulant, like they were in hospital, but they were not completely bedridden. Of course, they were not as physically active as they were before hospitalization, but they were not completely bedridden. So I can't really rule out the effect of less physical activity or more sedentary lifestyle or its contribution to the blood CAF22 levels. But like I see some effect even in those people who are symptom free, and they are of course quarantined, but they have some sort of activity, so they are not completely bedridden.